what are the uses of it. Now, did you people have any doubts in that?
No, ma'am.
Okay. Now, the next thing that we are going to discuss is molecular diagnosis. Now, students, tell me one thing. What happens when any one of you falls sick? When you go to the doctor, what is the first thing that is done? Also, everyone, switch on your videos.
Ma'am, I won't be able to switch mine on today.
Why? What happened?
The Wi-Fi is a bit weak.
Okay. Yeah, so tell me, like when you go to a doctor, if you have any disease, what is the first procedure that's done? Apart from taking
They
BP and all.
check the back of your throat.
Hmm?
They check the back of your throat
They check the
with
back
the
of your
stick.
throat. Okay.
With the
We
Yeah.
tell,
stick.
we tell the problem what we are having.
Yeah, you tell the problem. Now, what does the doctor do or suggest? Don't they ask you to go for a blood test?
Yeah.
Isn't it? To
Yes.
check the presence of pathogen. So that is a diagnosis, right? That is a molecular level of diagnosis, like what is happening in the blood cell, if there is any pathogen dividing or anything of that sort. So what is a diagnosis? See, diagnosis, it is a procedure wherein we determine what kind of nature of the disease is, what cause, like what is the causative organism of the disease, right? But you people know that you go to the doctor whenever you have any symptom present in your body. Any symptom, meaning if you have a fever, if you're feeling weak and all of that thing, right? So you go to the doctor only and only when you develop a symptom. So during the time of disease, okay, during the time of disease, what happens is that the presence of pathogen can be suspected only when the symptoms are produced, right? So during the time of disease, the presence of pathogen, now this pathogen can be anything. It can be a bacteria, it can be a virus, it can be a worm, etc. Anything, right? So the presence of the pathogen is detected whenever symptom is produced. Meaning during this time, if the symptom are produced, it means that during this period of time, the pathogen has, you can say, multiplied a lot in the body, isn't it? Then only the symptoms are being shown, then only you are affected by it, right? So the concentration by this time, the concentration of pathogen is like increased a lot. So the concentration of pathogen is, or you can say, has been increased a lot. Has been increased a lot. Now, when we talk about the methods of diagnosis, let us see what are the various methods of diagnosis. Now, if you divide into two types, right? One is the traditional, the old age methods that have been going on ever since time immemorial. And then there are some modern methods. So we are going to see what are the different kinds of methods of diagnosis. Now, the first one, when we talk about the traditional, so here what happens? Blood analysis, right? Blood test is done. So under the traditional, blood analysis. What else? Tell me. What else? Like the conventional, the basic methods of diagnosis. What are they? No idea. Apart from blood analysis, there is serum analysis. Right, this is like the routine procedure, right? That they with the help of injection, they take out your blood and they go like send it to the lab for testing. Apart from that, there is also urine analysis, urine and stool analysis. This is how the conventional method is. Now, do you think it is a very like the kind of method wherein early detection of the pathogen can be done in this kind of method? Do you think it is? So in this,
I didn't
it's not,
understand the
right?
question.
Hmm?
I didn't understand the question.
The question is, if we rely on the traditional method of diagnosis, do you think early detection of pathogen is possible? Early means much before the pathogen has not divided. Is it possible? Hmm? Did you get the question?
Somewhat.
It's not possible because, like I told you, whenever we go for the diagnosis procedure, the diagnosis is only done when the symptoms are produced. And the symptoms are produced when the pathogen has already divided and increased a lot in our body. So you can say in this, the early detection of pathogen or any disease is not possible. Isn't it, students? The early detection of pathogen or disease is not possible. When we talk about the conventional or the traditional method of diagnosis. But when we talk about the molecular or the modern method, so there is the first kind, that is recombinant DNA technology. We can use recombinant DNA technology for diagnosis also. Okay, so the first one is this, meaning RDT. Apart from that, there is also PCR. What is the full form of PCR, students? Hmm? The full form of PCR? No one remembers.
Polymerase chain reaction.
Polymerase chain reaction. This is also used for diagnosis in many areas, PCR. Then apart from PCR, there is a new thing that you are going to learn today is enzyme linked immuno enzyme linked immunosorbent assay. Sorbent assay. And in the short form, it is known as ELISA. Or ELISA, whatever you want to call it. To remember it, it is enzyme linked immunosorbent assay. And you can say now, in the modern method of diagnosis, in the modern method of diagnosis, early detection of pathogen or early detection of disease is possible. Early detection of pathogen or disease is possible because now we are playing at the gene level, right? And whenever we are at the gene level, we can see if there is any problematic area, if there is something that is going to trouble the human being, all in all. So in this case, the early detection, the early diagnosis is possible. So these methods are clear to you all, students. We will discuss the new methods in depth. Don't worry about that. But up until now, is everything clear?
Yes, ma'am.
Okay, very good. Now, when we talk about PCR, okay, PCR for diagnosis. Now, all of you have studied about PCR, right? I have only taught you this. So, talking about the PCR for diagnosis, first of all, What it does is that it helps to detect even a tiny level of concentration of bacteria, meaning during the time where the symptoms are not at all produced. At that time also it can detect the concentration of bacteria or concentration of virus. Okay, it can detect it so easily. Now, many a times PCR now is routinely used to detect HIV in suspected AIDS patient. You might remember RT-PCR, it was used to detect the coronavirus also. So it is a very powerful technique to identify many genetic disorders also. So PCR is that way. So when we talk about the PCR, so we can write in this that it helps to detect very low concentration of bacteria and virus, or bacteria or virus bacteria or virus much before the onset of symptoms, like much before the symptoms are produced, the disease can be detected, or the bacterial presence or the viral presence, it can be detected, right? And why and how actually? So by amplification of their nucleic acid. We know that during the process of PCR, multiple copies of a DNA can be made. So the bacterial DNA or viral DNA can be made into multiple copies. So if the multiple copies of the bacterial DNA is made, then that will tell us that, okay, the bacterial or viral disease is about to happen. Apart from that, you should know that PCR is now used to detect the HIV in suspected AIDS patient. What is the full form of HIV, students?
Human immunovirus.
Correct. So it is used to detect that. Now, the basic principle that goes behind this is that the DNA from the infected human or the suspected human will be taken, and it will be made to undergo PCR, that is several steps to make 1 billion copies, right? And if the viral DNA is present, so thousands and billions of viral DNA will also be copied. So the person, if a person has the virus or not, can be detected. Now, apart from that, one more unique thing is that the PCR can also be used to detect mutation in the genes, particularly for cancer patients. So, PCR can be used to detect mutations in genes in cancer patients. So, it can be detected, like it can be, you can say, used to detect the cancer in some people also. Okay, so this is like for many cancer also and many genetic disorders. So whether it is a bacterial disorder, viral disorder, cancer, HIV, all of these and many genetic disorders can also be detected by the help of PCR. Is this clear, student?
Yes, ma'am.
Yes,
Any
ma'am.
doubts? After that, we are going to see ELISA or ELISA for diagnosis. Now, what is the full form for it? It is enzyme linked immunosorbent assay. Now, this technique is based on antigen antibody reaction. Okay, it is based, like the principle for it is antigen dash antibody interaction. How that interaction or reaction takes place, right? What is an antigen, students? Tell me, what is an antigen? No one remembers. Antigen.
Um,
Tell me.
a foreign, um, a foreign substance
Hmm?
which may enter your body.
A foreign substance. What are you saying? Say it clearly.
A foreign substance which enters your body.
That is what? Antigen or antibody?
Antigen.
Yes, exactly. It can be any foreign substance, even if it is dust, or even if it is something bigger than dust, but non-living thing, that can also be considered as antigen by the body. And against the antigen, the antibodies are produced. Very good. Siddhika, you said the answer, right? Or Amna, who
Yes,
said it?
ma'am. Siddhika.
Okay, very good. Now, so that is the case, right? Antigens are those, antigen can be a living substance, it can be a non-living substance as well. So now infection by pathogen can be detected by the presence of antigen, meaning if antigens are present in the body, then that can be detected, or by detecting whether antibodies are made against that. Okay, or detecting the antibodies. Okay, so these are the things that can be used either during the ELISA or ELISA. What can happen is like either we can test the presence of antigens in the body, or we can like test the presence of antibodies that are synthesized against the pathogen. So by these two methods, we can easily identify whether you can say the particular disease is there or not much before it has multiplied a lot. So now here what happens is that the antibody is multiplied. Okay, and we know that antigens, they start, or you can say they attack the antibodies. So with the help of that, we can easily detect. Is it clear, students?
Yes,
Yes, ma'am.
ma'am.
All right. Okay, now you people can note down till here, then we'll move forward. You had to write some part from gene therapy also, isn't it? Or is that whole thing completed?
Completed, ma'am.
Completed by everyone?
Yes, ma'am. Done, ma'am.
Everyone's there? Students, all of you are done or not?
Done, ma'am.
Done, ma'am.
Everyone's done?
Done, ma'am.
Done, everyone?
Done, ma'am.
All of you are done, students? Tell me. Yes or no?
Done, ma'am.
Done, ma'am. Done, ma'am.
Okay, everyone's done. Any doubts up until now, students? Everything is clear, yes or no?
Yes, ma'am. Yeah.
All right. Okay, so now we'll move on to the next topic, that is transgenic animals. Transgenic animals. Now, these are all those animals that have their genes manipulated in some way, meaning they either contain an extra piece of gene or they do not contain the extra piece of gene. So talking about transgenic animals, these are the animals that have their genes, or you can say more DNA manipulated. Manipulated. Okay, they have their DNA manipulated to possess, so that they have to possess and express an extra DNA. To possess and express an extra, or you can say foreign gene. Okay, so these are the animals that have their genes or their genome or DNA manipulated to possess extra, or you can say to express that extra gene. Now, they can also be called as genetically modified organisms, right? Now, talking about the examples of all those animals that are transgenic. So, examples of transgenic animals. What do you think would be the example of transgenic animals? There are rats, there are rabbits. You might have seen on the back of the products that you use, it is written cruelty free, and there is like a rabbit made over there. It means that these products have not been tested on animals. So that rabbit can also be used as a transgenic animal or the animal for tests performance. Then there is pigs, they are also used. Then there are sheep. I'm not talking about animal this thing testing and all. It's just that these animals have had their DNA modified. Okay, and in higher organism there was monkey. There's also fish. But if we see, all in all, out of all of these, remember that 95% of all the transgenic animals, which one do you think is the most common, students? Hmm? They are mice. So 95% of all the transgenic animals are mice. Rest all come under that 5%, but 95% of them are mice. Now the whole entire question comes along, that how do we make the transgenic animal? So we make the transgenic animals by incorporating, or you can say by RDT technology, isn't it? Recombinant DNA technology, wherein we transfer the gene of interest, we do DNA micro injection, isn't it? And why do we produce them? Some transgenic animals, they are produced for specific economic traits. So the question comes, why do we produce, like what is the reason that we produce the transgenic animal? So talking about why do we produce it, so the first reason for producing it would be some transgenic animals are produced for specific economic trade. Some transgenic animals are produced for specific economic trade. Meaning anything that would generate income. If there is any character or any trait that can or has a potential of generating an income. So that is why we can produce the transgenic animal. Now, the other transgenic animal, the other transgenic animals, they are produced as disease model. Meaning, for example, if if there is like a scientist and he or she is studying any particular disease, so he can if he wants to see how the disease works or which drug would affect on that, which would not, so they are produced as disease models, meaning we test our disease drugs and all on these animals. So that is another reason by which this is produced. Now see, the transgenic animals can benefit us under three major categories, just like how we studied in this chapter: medicine, agriculture, and industry. These are the various ways by which they can say benefit us. So benefits from transgenic animal are produced, or you can say are under three broad categories. What are those categories? The first category is that we can benefit from them in terms of medicine. Meaning not just medicine means drugs. No, medicine not only means that, it means something high in nutrition or something beneficial in terms of our health. The next is beneficial in terms of agriculture, how they are beneficial over there. And the third one is beneficial in terms of industry. What can we do by making mass production from them, right? So the first case is medicine, the second is agriculture, and then there is the industry. Now, when we talk about the medicine, here the cows that produce, example is cows that produce human protein rich milk. Human protein rich milk, meaning the nutritional value of that milk is high, and that is a cow. The cow has been, you can say, made transgenic, that it is producing a superior product. Then for agriculture, you can say sheep that grows more wool, that grows more wool. And then talking about the industry, so one such is boats that produce silk. This is one such example. Okay, so like in all of these industries or in all of the three categories, there are certain examples that can be taken. These are the examples. Now moving forward, let us see in under the field of medicine. Okay, so we can use the transgenic animals to study the normal physiology and development. to study diseases, to produce like biological products, to test the vaccine safety. Also, chemical safety testing can be done, right? So, all of these things can be done as far as the particular medicine aspect undergoes, right? So, under the medicine category, under the medicine category, the transgenic animals can be used for the following. Let us see what are they used. Now, the first, like I told you, to study normal physiology and development. To study the development and physiology. Then it can also be used to study diseases, right? Various, like they can be disease models, like I told you. Then they can be used to produce biological products. Biological products. Now, what are the biological products? For example, there is a particular, you can say, transgenic animal that can be used to produce alpha trypsin, or you can say alpha 1 antitrypsin, that is used to treat emphysema. Similarly, there was the first transgenic cow whose name was Rosie, and like that cow was responsible for producing human protein enriched milk, meaning the milk with high nutritional value, right? And that can be used for human babies. Okay, that is like more beneficial than the normal cow milk. So the biological products under that, alpha 1 antitrypsin, for treatment of emphysema. Emphysema is the name of disease. So for the treatment of emphysema, okay. And the attempts now, in 1997, the first transgenic cow, named Rosie. Her name was Rosie, was made. It produced protein enriched milk. Meaning normally what happens is that in the cow milk that we drink, it has more fat and less protein. But now the milk that was produced by Rosie, it had more protein and less amount of fat. Okay, so that is how. Now the fourth use in the field of medicine is vaccine testing or vaccine safety testing. We know that before the vaccine is released into the market for human being, the mice, they are used for the testing of their safety. Then comes the chemical safety testing. Chemical safety testing, meaning if, for example, there are certain drugs, there are certain like anything, any food product, any of those things that are released into the market, they are checked with the help of these transgenic animals. Any doubts up until now, students?
No.
Okay. Now let us see how the use, or you can say under the particular agricultural field, under the agriculture category, the transgenic animals are used as are used for, let us see what are those reasons that it is used for. The first one is for breeding, right? For breeding, meaning many, or you can say there are many plants or animals that have the desired traits. So we can use them in the field of breeding. Second is for quality, meaning like high quality nutritional value products can be made. And lastly, to test the disease resistance. So these are all the ways. Okay. Now you people can note it down. Everyone's done?
Yes, ma'am.
Done.
Done, students? Done? Everyone's done?
Done. Done, ma'am.
Done, students?